Welcome students. Myself Shaila Shetkar, Assistant Professor in Botany from Government College of Arts, Science and Commerce, Sakli. The program is Bachelor of Science, first year. Subject is Botany, generic elective. Semester is 2. Course code is BOG 102. Course title is Coastal and Mangrove Ecology. Title of the unit is Biology of Mangroves. Module name is Factors Affecting Various Growth Parameters in Relation to Physiology and Biochemistry. This is the outline of the module. In this module, we are going to learn about the physiological and biochemical factors affecting the growth of mangroves. These are the learning outcomes of the module. At the end of the module, you will be able to describe the physiological and biochemical factors affecting the growth of mangroves. Coming to the introduction, the mangrove adaptation in the environment such as salt tolerance ability are essential for survival and development of mangroves. In an unfavorable condition, the adaptations of mangroves such as aerial roots and salt excreting glands present in leaves makes them perfect natural models to cope with various abiotic stresses. Mangrove trees contain roots with higher extent of gas freedom, while waterlogged are filled for salt tolerance and salt regulation. The salt and soaking adaptations of mangrove is one kind of morphological and physiological trait for living in the tidal condition. Now let us learn about the factors affecting growth of mangroves in relation to physiology and biochemistry. The physiological factors are salt concentration, photosynthesis, transpiration and tidal fluctuation. Biochemical factors are phytochemical constituents, pigment concentration and nutrient availability. Now let us learn about the physiological factors affecting growth of mangroves. First is salt concentration. Growth rate is maximum at an intermediate value of salinity and is lower in more fresh or more saline condition. Increase in water salinity decreases shoot and root biomass of mangrove seedlings. The nitrogen assimilation rate of mangrove seedlings also decreases with rising salinity. Mangroves regulate salt by excreting it through salt glands. As water salinity increases, some species become highly conservative in their water use, thus achieves greater tolerance. If the water salinity is very high, the mangrove species need lot of energy to overcome the high salinity level. Mangrove trees are salt tolerant species, but if they take more salt from the water, then they need lot of energy to excrete the excess amount of salt from the plant body. Therefore, if the water salinity remains high, mangrove exhaust themselves and their growth will be stunted. The next is photosynthesis. Photosynthetic rate of mangroves is strongly affected by environmental conditions. Mangrove species are excellent at absorbing and storing carbon from the atmosphere. As the tree grows, they take carbon from the atmosphere and use it as building block for leaves, stem and branches. Low salinity conditions reduces carbon loss, which leads to a greater CO2 assimilation. Thus, fluctuating soil salinities can reduce photosynthetic rate, which may lead to a stunted growth of mangroves. The next is transpiration. Transpiration is one of the major factors that affect the growth of mangroves. Some xerophytic features of mangroves like thick cell of stem and petiole, thick cuticle in leaves, sunken stomata, multi-layered epidermis, Sclerenchymatous bundle sheath helps to control transpiration rate. Thus, they conserve water by regulating the loss of water vapor by transpiration in a saline environment. Next is tidal fluctuation. Tidal fluctuation plays an important role in transporting nutrients, exporting waste, and effective dispersal of propagules, which leads to a productive growth of mangroves. When evaporation is very high, tidal fluctuation washes excess of salt away, preventing high soil salinity. Now, let us learn about the biochemical factors that are affecting growth of mangroves. The first is phytochemical constituents. Mangrove grows in solar radiation environment that are high in UV radiation. High levels of UV radiation generally reduce plant growth rate and photosynthetic activity. Mangrove leaves are rich in phenols, polyphenols, tannins, flavonoids which act as UV screen compounds. Hence, mangroves tolerate solar UV radiation and create UV free under canopy environment. The next is pigment concentration. Chlorophyll and carotenoid levels are high in mangrove during the summer, which increases the photosynthetic rate. Anthocyanin acts as photoprotective pigment. The concentration of anthocyanin is highest in the monsoon season, whereas flavonoids increase in pre-monsoon season. Anthocyanin also plays an important role in reproduction by attracting the pollinators and seed dispersal, and also protects them against various abiotic and biotic stresses due to their powerful antioxidant properties. The next is nutrient availability. Nutrient availability is one of the major factors that affects the mangrove forest structure and productivity. Mangroves are deficient in potassium and nitrogen, which limit their growth. Therefore, microorganisms like phosphate solubilizing, nitrogen fixing microorganisms, and mycorrhizal fungi solubilize the bound phosphate into available form. These are the references of my module. Thank you.